Hello all. So in this we will be learning how to do a sequence analysis. So in this video we will be learning how to use tools like Ncbi Blast in order to find out whatever our sequence could be. So I have a worksheet where we are having hold on, I will share my screen. So I have a worksheet, so where you have a sequence that we need to analyze. So I am copying this sequence. This sequence is in the FOSTA format. FOSTA format refers to any sequence that is having a forward arrow mark and some information about it in the first line. And from the next line, your sequence will continue. So this is because whatever the tools that you are using should be able to identify what is the sequence and what is the information. So the next thing we are going to do is go to Ncbi. So in Ncbi, so this is one site where you can use a number of tools, and it is basically a database that is having DNA data, RNA data, about genes, genomic data, SMP data, and everything. So this Ncbi has a tool called Blast. So these are all the 4 types of blast that Ncbi is going to provide you. In case if you have a nucleotide sequence that has to be analyzed, you will go to nucleotide blast. And in case if you have a protein blast that has to be analyzed, you will go to a protein blast. So since we have a nucleotide blast, let's select the nucleotide blast. So this is how the page opens, and this is where you will have to paste the sequence. And you need not change anything. So because the worksheet, if you go back to the worksheet, it says that this following transcript was found to be abundant in a human patient's blood sample. So transcript is something that you will be getting after transcription. So that is, it says that it is an mRNA. So this happens to be mRNA that is found to be abundant in a human patient's blood sample. So that is the reason when we are looking at the database is searched across the nucleotide collection. The moment you select blast in, it will be searched across the nucleotide selection. And if you are specific, like you need to look under a certain organism, you can enter it there. So because we know that this nucleotide belongs to humans, we will give Homo sapiens. So tax ID is there. Just click and you give add. So this is added, and then just blast. So it will take a couple of times for your result to come. So basically what the blast is going to do is it is going to search your query sequence, that is a sequence that you want to analyze, with every other sequence that is there in the database. So this is how the result comes. Let us look at the first 4 hits. So everything has a query cover of 100%, and percentage identity is none of them are showing 100% identity, which means that the sequence that we are searching is not deposited in the database. So that data is not there in the database, in the nucleotide database, but there are some sequences which are similar to what we are searching. So the first hit what we have got is Homo sapiens hemoglobin subunit beta. So when you click on this, it will take you to the alignment. So if you look at the description, it is having something called as the accession number, with which it is identified in the database. So if you click on the accession number, it will take you to the particular page in GenBank. It will take you to a particular page in GenBank where it has every other detail about this sequence that you are trying to search, like what is its locus, on which on which part of the chromosome it is located, what is the definition, what is the accession number, everything, organism, and in what all journals this sequence have been published, and from where this data comes. So it also has a summary. So the alpha and beta chain loci determine the structure of type 2 polypeptide chains in adult hemoglobin and every other data. So through this, we got to know that the sequence that we are having is similar to hemoglobin chain beta, but because it is not 100% identical, we can tell that this is a mutant beta globulin. So if you go back to the blast and if you click on this, it will take you to an alignment page where your query is aligned with the subject that it has found out. So this first line is the query, your sequence, and second line is the original sequence of that beta. So wherever there is line, it is telling that the nucleotides are matching, but there are few regions where there is no line. Here and one more is somewhere. Yeah, one is here and another one is here. So the hemoglobin beta chain is having adenine, but in our sequence, it has got mutated to thymine. So here in the original sequence, there is thymine. In our sequence, it has got mutated to adenine. So when we look at this mutation, these are point mutations. So if you look at the summary, it says that mutant beta globulin causes sickle cell anemia. So if there is any mutation in this chain, it leads to sickle cell anemia. With this, we can conclude that the sequence that we are having is that of the sickle cell anemia. So the patient is suffering from sickle cell anemia. From here, we can go to gene database. So here you find all other details about this gene, like what is the chromosome, and in this chromosome, in this loci, where is the gene? So this blue line is the chromosome background, and wherever the green line is starting now, so that is where your HBB gene is present. And it also gives all other details about this gene. There is one more website called, a database called OMIM. If you go to that, before that, so in the gene, you will have what is the official symbol, full name, primary source, gene type, all other details about this. So when you go to OMIM from GenBank, so it will take you to hemoglobin beta locus. So this is where you can get details about the disease. So what are all the diseases that could be possible because of any changes in beta chain is listed in this table, and description regarding the same, like what is the gene structure, how is it mapped, every detail, what is the gene function, everything will be listed in this. So this is a short snap of how to use the blast. So now we got to know that whatever the sequence that we are having corresponds to hemoglobin subunit beta. So in order to compare that, we will be using a tool called multiple sequence alignment, which will be shown how to use in the next video. But before that, how to reach to the original sequence if you want. If you want to know what is the original sequence, simply go to NIH and type HBB. We know that the name of the official name of this gene is HBB, which is the original gene, for which we all have a mutant one. And here you select nucleic acid database, means you're asking it to search within the nucleotide database. And when you ask it to search, the first thing that you're going to get is result about HBB hemoglobin subunit beta. So if you look at this reference sequence transcripts, this is what is your sequence for original beta and mRNA sequence, and this is your protein sequence. So both of them you can download here only. So just click on this and you will get subunit beta. Go to FASTA, copy this, copy, make a folder in your drive. Make a folder in your drive. Just say, I will save it as hemoglobin beta analysis. Okay. So here, go to create a new text document. This is original um, original nucleic acid, original mRNA for hemoglobin beta. Correct. Open this and paste this. No, I'm so sorry. Everything has got selected. We'll go back to this one and we'll just copy this one. And we'll go to the text file. We will paste it. So this is the header, right? So I'm removing this information and just writing original mRNA HBP. Okay. And save. Just close it. So in your folder, you have original mRNA HBP. In the worksheet, you will have the sequence that you are using to search. Right? You can copy this also and put it in a text file so that it is easy for you to analyze. Just copy, go to your folder, new text document. This is mutated mRNA HBP. Okay. So open this and paste this. So I will rename this also header as mutated mRNA HBP just to avoid long names. That's all. It is asking to say just save it. So here you have original mRNA and mutated mRNA. So this is how you will get sequences, mRNA sequences for original gene where your protein belongs to or nucleic acid belongs to, which you will be using to compare.